Hi everyone, my name is Ziynet. I'm a PhD candidate at University of North Texas and also research assistant at Dr. Serdar Bozda's lab. Today I would like to present Supreme, a cancer subtype prediction methodology integrating multiomics data using Graph Convolutional Neural Network. Cancer patients show different characteristics in terms of progression of disease and response to treatment. Breast cancer is the most commonly diagnosed cancer worldwide. There are some biomarkers in breast cancer which guide to different treatment decisions. In spite from that, researchers studied molecular subtypes to find different characteristics in cancer. In 2009, Parker et al. Find that expression of 50 genes was important to cluster breast cancer subtypes, which are called pan50 subtypes, and concept as granted for many studies. With increasing technology, various biological data sets from cancer tissues have been generated. And for instance, the Cancer Genome Atlas project generated data for thousands of patients for 33 cancer types. In TCGA, there are 7 different multiomics data to better characterize breast cancer, considering that each of them shows a unique aspect of underlying biology. It's important to integrate them. So this is called as multiomics integration. There are some studies for cancer subtyping. Some of them are doing some integrative multiomics integration, doing some dimensionality reduction, utilizing some patient-similarity networks, or doing some data perturbation. Some of them are utilizing gene expression with pathway and utilize some deep learning. And also graphs are very important to store some information. Graph representation learning, those are trying to represent a graph in a meaningful way, and GNN is the recent concept in that one, and GCN is one of the popular one. It just simply average the neighborhood information, including the node itself, and applies neural network for that. And there are some GNN-based approaches like GCNC, MogaNet, which utilizes single network or multiple network, and they are trying to utilize those GCNs. However, to utilize more from available knowledge, it's important to properly integrate multiple networks and multiple features at the same time. For that, we introduced a node classification framework, Supreme, which is a cancer subtype prediction methodology. And it does a graph convolution on multiple patient-similarity networks. And on those networks, we have multiomics features encoded. And this tool will be publicly available soon on GitHub. And first, we collect data and we extract features from each data type. And we are showing that each row is a patient in that matrix. And we build patient-similarity networks from each data modality separately. Our assumption is that if the 2 nodes are similar in terms of local neighborhood, then they will be belong to the same subtype, where the similarity is based on one specific data type's feature. Here, blue data type features are used to generate those patients pairs for those edges, and for the others as well. And we compute patients embeddings, and we train a GCN on each network separately, and we code all the multiomics features and all the extracted features as raw feature, and we utilize all the raw features as our node features on GCN. To extensively evaluate our performance, we run each combination of embeddings as a separate model. We have 7 embeddings, so 2 to the power 7 minus 1 different models. And we integrated raw features to each embedding combination in case the signals were diluted while we are doing convolution. And then we did cancer subtype prediction on a multi-layer perceptron. And we apply Supreme to TCGA, Metabaric, and the combined one separately. And here the task is cancer subtype prediction. And we are using grant root as pan50 subtypes, which are readily available at those data sets. We will show results on TCGA only for the sake of time. And here we show the performance of Supreme against the other tools. We use macroinfant score as our evaluation metric. Since we have 127 different models per method, we use the violin plot to visualize the overall performance. And we also ran Supreme without raw feature integration to see the impact of that step. So this is called supreme minus. And according to our results, supreme and supreme minus outperformed other tools. And also, even though there is not a significant difference between supreme and supreme minus, supreme outperforms supreme minus for most of the cases, showing the importance of raw feature integration. And we wanted to measure the ability to differentiate samples based on survival too. For that, we obtained p-values. Here we are showing those p-values for each method, showing the significance level of survival differences between subtypes. Here again, we have 127 different models. So we had 127 different p-values. And the red line shows significance level, the dashed line shows grand truth p-value, and the results show that supreme had consistently significant differences in survival rates and outperforms other methods significantly. And supreme p-value was mostly better than grand truth significance level. In conclusion, we developed supreme cancer subtype prediction methodology by doing convolutions on multiple patient similarity networks. We showed that supreme outperformed other tools in terms of cancer subtype prediction and also in survival differences. For survival results, we saw that it was better than grand truth, which is based on gene expression data type. This might suggest that supreme could demystify some undiscovered parts of the cancer subtypes, and utilizing more knowledge from other data types, we could improve the grand truth label. And utilizing the recent advances like GNNs, we could have more clear cancer subtype groups. And this will help us to pave the way for precision medicine. I would like to express my special thanks to Dr. Serdar Bozdağ for his advice and supervision, and also my lab friends for their support. I would like to acknowledge our NIH grant, and also too many thanks to all ISMB organizers for this opportunity. Thank you all for your interest.